Great. We'll let people keep trickling in, but we'll go ahead and get started for now. It's so good to see everybody today. Thank you so much for joining us. We always appreciate you joining us for Paleo Perks. Throughout the seminar, you're welcome to send questions to Aharna, who is our questions host this morning. I'm sorry, I keep saying this morning. It's morning for me today, at whatever point. Um, and we'll go ahead and get started. So today we are thrilled to have Maria Hussain with us here from the University of Manitoba in Canada, and she's going to be presenting a talk called Irresistible Insights: What Can a Fish Ear Stone Tell Us About the World? Um, the format of today's seminar will be that we have the welcome and announcements, which is currently happening, then the talk for about 30 minutes, moderated Q&A for 10 minutes or so, and then tea time if you'd like to join us to talk to Maria and learn about her path and career, and chat more about research. And again, throughout the seminar, you're welcome to send your questions via chat to the questions at Paleo Perks host. Just some housekeeping today. We do value the participation of all folks interested in the paleosciences, whether you're a very passionate high schooler or a very tenured professor. Anywhere in between, you're all welcome to join us. We're happy you're here with us today. We just ask that everybody abide by our code of conduct that you had to sign to get today's link. You should automatically be muted, and we need to ask that you stay muted for the duration of the talk so we can hear everything Maria has to say. And again, you can ask questions by chatting to the questions at Paleo Perks host, which is Aharna this morning. And once we enter the Q&A, you're also welcome to use the raise hand function. We do have closed captions built into the Zoom, so they should be showing up automatically, but if not, you should be able to toggle that on. And we'd love for you to nominate an early career researcher. We'll be dropping the link in the chat. And if you have any ideas of who you think should be giving a Paleo Perks talk, please let us know, and we'd love to hear your nominations. At the end, we'll also be giving you a weekly feedback form. This is where we ask for demographic info just to find out who's attending the seminars and who isn't, if there's anybody who isn't being included by any of the current policies. We want to make sure that they're as accessible as possible to all who are interested. Also, we're currently discussing whether we should perhaps change the frequency of talks to every other week. If you have any feedback on that in particular, if that would be too confusing, then we would be happy to hear that. Do you like having it every single week and just knowing exactly when it's going to happen, or do you have other preferences? With that, we're going to turn it over to Maria Hussain, who did her bachelor's at Thompson Rivers University in Canada and is now a master's student at the University of Manitoba. Maria, with that, it's off to you.
Hey everyone. Yep. Okay. Let's try to see if I can get my thing set up again. Thank you so much for the introduction. All right. Hey everyone. So I'm Maria Hussain, as Gwen said. Today I'm going to give you a talk about fish ear stones, also known as otoliths. And yeah, let's dive right in. Oh, I forgot my little animation. That's me saying, I love dissecting fish to study their ears. And then that fish goes, what? So, hey, Paleo Perks. I'm a first-year master's student in the earth sciences at the University of Manitoba, co-supervised by Dr. Kirsten Brink and Dr. Michael Schindler. Kirsten Brink is a paleontologist, and Michael Schindler is a mineralogist, geochemist, something along those sorts. I will discuss my background a little bit later, but I came from a biology side of things. So yeah, it's a fun mix of things. I also work as an aquatic science technician at the Government of Canada in the Department of Fisheries and Oceans. That's what I do for work. Okay, some fun facts. So like I mentioned, my background was in biology and specifically aquatic ecology and limnology. So I had this whole idea in my head that I was going to be a limnologist or some kind of ecologist that only studied water. I had a perfect professor in mind. And then when I asked him if he was taking on any students, he said, I'm retiring. And I said, can you stop? And he said, no, I'm going to retire. So he retired and I was left like, what do I do? So in my last year of undergrad, I started looking for next steps. And at that time, I was actually in a paleo class with Kirsten. And just got to talking and I was like, hey, so paleoecology is a thing. Like I always liked, I'm gonna be a big millennial moment. I loved dinosaurs. I liked Jurassic Park. What can I say? But I didn't know much about paleo. So as I took that class, I got really interested in paleoecology and how you can do ecology of today's date, so modern day, but you can also do it of the past. And just the idea of how you could possibly tell interactions of something that happened so long ago was so intriguing to me. Um, so from there, we kind of started discussing a master's program, and she had this project about fish ears, and I was like, okay, sure, that sounds weird and awesome. I didn't know much geochemistry. I didn't know much about biomineralization, but I learned a lot. And so, yeah, that's where I am now. A couple other fun facts. So my COVID hobby was making stickers. It was really fun, but I made some nice sciencey stickers. I made some bookish stickers. If you aren't interested, scan that QR code and you'll be taken to my little Etsy shop. And then my very first conference is this picture here. That was in 2023, and it was an international conference all about otoliths. I was I was so surprised. I was like, okay, sure, little old me in an international conference. At that time, I was still in undergrad, and I was like, okay. So I went, I made this gorgeous poster. I was so proud of my work, and it just so happened that I won best poster. Um, so honestly, I was like, oh my God, academia is for me. Because it was just such a, like a pat on the back. I feel like I've been good at things, but I never got an award for it. So I was like, all right, sure, I'll be bashful and yeah, I'll take it on. All right, so let's discuss my master's research. My master's research predominantly focuses on mining related pollution effects on otolith biomineralization, with a specific emphasis on zinc contamination, because my study site, which is in Flin Flon, Manitoba, and I know not a lot of people are aware of Manitoba geography, so that's in northern Manitoba. I believe there's 3 definitions of what an Arctic area is, and that falls into the tree line definition. So parts of Flin Flon are considered within the Arctic, not the circle, but you know. So it's a really remote, not that remote. It's pretty remote, and it's a northern community. There's a mine out there that's been running for the last 90 years, and they were mining for zinc and other forgetting the word, but other metals. Uh, we yeah, like I said, we study otoliths, and I will discuss what otoliths are in just a second, but I wanted to introduce my project a little bit first. So the industrial processes that have released these heavy metals, including zinc and selenium, into the local waterways have been raising concerns about the impact on aquatic organisms. So therefore, there are 3 main research goals for my research. One is to determine if zinc levels in otoliths reflect levels in the water and in the sediment. So is what's being uptook, whatever uptake is happening, is the bone reflecting the levels that are in the water column or in the sediment? Number 2 is to determine whether biomineralization is affected by trace metal uptake or changes in the elemental availability in the water. So if different times of month or different years, there is more or less zinc, all questions I'm really curious about. And number 3, this is where the ecology comes in, is to determine if different trophic levels impact the amount of trace metals found in otoliths. So does the big fish that eat a bunch of little fish have more zinc, or does the little fish have more zinc? Because the little fish was eating the sediment. We'll find out. My research has a lot broader implications than just Flin Flon or northern communities or lakes or pollutant lakes. If we can understand how pollutants alter otolith formation, we can use these structures as bioindicators to monitor water quality and fish health in other contaminated environments. So I'm looking at lakes, which are freshwater, but we can use the same methodology to look at marine fish or brackish fish, right? This could help guide conservation efforts and inform environmental policies aimed at mitigating pollution effects on ecosystems. So I keep throwing around the word otoliths. So what exactly are otoliths? Where are otoliths? let's go into it. Otoliths are calcified structures found in the ears of all bony fish. So any fish that has bones has ears or has otoliths, right? This diagram shows a wonderful area where they're kept. So they're right behind the eyes, or like, just like our ears are right behind the eyes. And there's one on each side, and each otolith sits in like a little fluid-filled area, and they are like that because they help with balance, hearing, and movement. So otoliths, these three pictures down here, we have two whole otoliths of different species. As you can see, they look completely different, but they are both fish. One is an Arctic cod, and then the other one is a Greenland halibut. And then I have the other picture on the far right here, and that's a sectioned otolith. So usually when we start looking at how to study otoliths, we will transversely section them, and then we read the section to age them. So otoliths are calcified structures composed of calcium carbonate and usually aragonite. More on this later. And there are kind of two areas so scientists will either pick aging for otoliths or microchemistry. I kind of do both. So in my work, I work in an age lab, and we focus a lot more on the aging side of things. And then for my master's, I'm clearly doing a more microchemistry master's. So looking at incorporation and biomineralization. Now, the interesting thing about otoliths is as fish grow, they form growth rings, but these otoliths also contain information that's recorded in those rings. So if there's a month where a bunch of zinc was released in the water and that fish was actively growing and it sucked up all that zinc isn't going to go anywhere. It's going to be stuck in that otolith. So even if I look at that otolith 30 years from now, I can still see that 1 year that there was a lot of zinc. As fish grow, they form these growth rings, and yeah, it holds on to that information. So you can tell a lot from an otolith, and I will discuss a little bit later what we can tell. These growth rings allow researchers to gain insights into the history of the fish, their movements, and even environmental changes over time. They're useful for fishery science, particularly in managing fish populations, studying migration patterns, and they also hold potential for environmental studies of the past and the present. Now, let's talk about how otoliths form. As mentioned, otoliths are made of calcium carbonate, which is sourced from both the water and the food that the fish eats. The rate and pattern of growth vary with metabolic processes. So in the warmer months, otoliths grow rapidly, forming translucent rings. And while in colder months, they grow more slowly, creating a dense dark ring. These annual growth rings can be used to estimate the age of the fish, and I do have an example of that here. So each red dot is where you would say a year is. Now, as you could see, in the first 5 to 6 years, they're pretty far apart. They're really nice and spacious. And then like the last 4 years, they're all condensed together. And that's because the fish has matured and it's growing. It's done growing pretty much, right? Like, so it'll still continue to grow, but it's gained all the weight that it needs. It's gained all the length that it needs. So there are some ways that we can kind of age them better. So if you age an otolith whole, where I think this one might be a whole otolith, if they look really, really close together, you can section it and then see if that clears up anything. You can burn the otolith and see if that clears up anything. It's a lot of trial and error, but there is, yeah, like that hundreds of people out there that do aging for a living. So yeah. The continuous accretion of calcium carbonate can also be disrupted by environmental factors such as water chemistry, temperature, and pollution. Understanding these patterns is central to my research, especially in the context of how trace metals like zinc are incorporated into otoliths. To analyze the otoliths at the necessary level of detail, we use several advanced instruments. One of our first steps is to use laser ablation and inductively coupled plasma mass spectrometry, LAICPMS. The ICPMS allows us to perform elemental analysis on otoliths at a micro and nano scale. This technique is particularly useful for examining the incorporation of trace metals into otoliths. Then, depending on what detail we want to see, we can use the scanning electron microscope to examine surface morphology and otolith structure, or if we want to go in even deeper at a nanoscale, we can use the transition electron microscopy to provide a high resolution imaging of the crystallographic properties of growth patterns at a nanoscale. I will show some examples of TEM images later on in this, so stay tuned. Together, these tools give us a comprehensive view of the composition and its relation to environmental factors. And then we go from there. So after that is where you start thinking of the ecology. So while I love doing the instrumental work, I really like the part after where I'm looking at the data and just holding my head and I'm like, what does it mean? So because I'm relatively new into my master's, I wanted to pull in some papers from the otolith web out there and just discuss how otoliths can be used in different aspects of research. Now, some examples of how otolith growth can be disrupted is this study by Lepke et al. I'm going to try not to say names. I'm pretty bad at pronunciation, but it highlights the effects that temperature and PCO2 has on otolith biomineralization. I'm mostly focusing on temperature, just because they found that the CO2 didn't actually have a giant effect. So temperature changes can shift the calcium carbonate polymorph in otoliths. So most otoliths start off as aragonite, and then they will sometimes turn into calcite, or sometimes they'll turn into vaterite. It's rare, it's very under-researched, and it's really, really curious, but I sadly don't have all the answers yet. But this study showed that with increase in temperature, a lot more otoliths were turning into vaterite than they were calcite. Now, normally, from what I've kind of read, calcite is the most stable version of calcium carbonate, but in otoliths, it just seems to be different. And sometimes what we visually see as vateritic, because there is an area where we kind of determine if it's vateritic or not. It just, yeah, sometimes it is, sometimes it isn't. But this study did show a really good way that temperature can affect and change the polymorph of calcium carbonate. So that's completely disrupting the biomineralization. And if we looked at that under the TEM, we'd see a lot more disorder. So yeah, this study didn't focus on heavy metal contamination, but the idea here is that, hey, maybe there was something metal uptakey going on that led to that change along with the temperature. Another study is actually one that I was involved in, and we looked at trace element incorporation in channel catfish otoliths. This is the research that I won my best poster award for. So yeah, this is the research that kind of allowed me to pick my passion and then keep going with it. So our first step, very similarly to what I discussed in my methodology, was to do an LAICP-MS scan. We observed that there are certain growth domains, particularly in the nucleus, which is like usually year 0 of the fish, that had high levels of phosphorus and sodium. So we wanted to, we were just like spitballing ideas. We were like, okay, well, let's just do some TEM prep on it and see if there's anything interesting there. When we did our TEM prep, we found that there were areas that showed little bunches of phosphorus and sodium, and that they were directly relating to the disorder that we're seeing in the growth domains. So figure E and F here showcase in the same area, different scales, but similar. So E shows within the aragonite matrix, there are areas where it's like darker and the lattice just starts growing jaggedy, and we're just not really sure why, right? So when we do a chemical map on those areas, we're seeing that it's directly correlating to high levels of phosphorus and sodium. Now, these irregular growth domains seem to correspond with areas of trace elemental incorporation. In other words, the presence of trace elements might directly impact how otoliths grow and form. We're still working to fully understand the implications of these distribution disruptions, And because these fish were from a river with a lot of phosphorus runoff, it's interesting that at a nanoscale we're seeing disorder, but the fish on a macro scale is completely fine. But if something is happening at a nanoscale, it's possible, like hundreds of years down the line, it could grow to be something more. Now, let's shift gears a little bit and look at the Western Interior Seaway, which I'm hoping that my paleo people are very, very comfortable with, because I find it an enigma. The Western Interior Seaway was a vast inland sea that covered much of North America during the late Cretaceous. Now, most studies of the Western Interior Seaway have focused on the western margin, whereas the northern and eastern margins have been relatively under-researched. Now, if I can, I'm not sure if you can see my mouse here, but that's kind of where Manitoba is, and that's the area that we're studying for this study here. It's right below the Hudson Seaway if you can't see, but maybe, yeah, we can discuss where Manitoba is later. Now, our lab, Brink Lab, has a couple of projects regarding the northeastern margin of the Western Interior Seaway. There are two studies that I want to discuss. One was a previous study on microvertebrate fossils that was, it was meant to investigate the changes in assemblages and environmental conditions. So Aaron Kilmarie in 2023 did some zonation work. I did some more research on this, and I will discuss that in just a little bit as well. Secondly is Bruno Costa, who's a PhD student in our lab, and he's investigating the lack of ammonite fossils, and more broadly, the lack of calcium carbonate structures in the Pierre Shale. So geological little time frame there, just so everybody is acquainted. Kilmarie et al. And my research was in the Falco Formation, and then Bruno is a lot later in the Pierre Shale. What's interesting is that the Falco Formation we were discussing, it has some forams, but we haven't found any otoliths yet. So, and Pierre Shale has nothing, so weird. So here's where things get a little interesting. A study I conducted examined the data that Kilmarie et al. Collected on microvertebrate fossils from the late Cretaceous in the Falco Formation. So figure A and B here. A, you can see that the blue area is a lot smaller. It's around 25% of the total. So this is a chart showing species occurrences in percentage. The blue area that I'm looking at is bony fish. So 25% of bony fish in the entire fossil record. The biggest area is reptiles, because we just find a lot of them. So Mosasaurus, Plesiosaurus. And then in chart B, we can see that that area grew from 25% to 36%. And that was directly correlated with us actively searching for microvertebrate fossils. We discovered a higher occurrence of small-bodied vertebrates, such as bony fish and birds, when microfossils were included in the analysis. This suggests a richer, more diverse, and more expected ecosystem capable of supporting the large reptilian predators like the Mosasaurus and Plesiosaurus. This shift in the fossil assemblage can tell us a lot about the paleo environment, including how different species interacted within ecosystems. So it just shows us that when we try to find the small fish, we can usually find the small fish. The absence of otoliths in the fossil record can be explained by several hypotheses. So the diagenesis hypothesis is most likely is that post-depositional chemical changes like acidic pore waters may have dissolved the otoliths or have caused them to recrystallize into other minerals, which eventually led to complete dissolution. Then there's environmental chemistry, which kind of suggests that low oceanic calcium levels or high CO2 concentrations could have impacted carbonate preservation. And then alternatively, it's like biological. Maybe the fish metabolism or water temperature influence how otoliths formed. And this led to, again, similar things. Like if aragonite could have been found, maybe it changed into vaterite or calcite and then it melted. So moving on to fossil otoliths, they do exist. I sadly haven't found them in my area, but they do exist. So this one study done by Velikoup actually just came out 2 days ago. So while I was looking up just last minute things, I found this study and I was like, hey, this is so cool. Why didn't I read this before? It's because it was released on February 2nd. So that's fun. This shows us that by analyzing stable isotopes in fossil otoliths and in shells and bivalves, they tell us completely different things. So in this figure, the fish down here is what shows you what the otolith signature revealed, and then everything else is all the other things. So what they found was comparing isotopic ratios of gastropods, bivalves, and otoliths. Otoliths showed a deeper water signatures of around 30 meters, whereas the bivalves and the gastropods showed 0 to 5 meters. So really shallow environment. This likely means that the otoliths were transported because when fish are eaten, the otoliths stay intact. So sorry. So what this really shows us is that otoliths can give us a much broader perspective of what the ocean climate looked like back then, rather than gastropods, which obviously do give us something and more accurately reflect the environment they were found in, whereas otoliths kind of drifted away and they're showing us a different environment. But it's intriguing. It's still cool to know that how much oxygen was found in the gastropod that's probably here, and then how much oxygen was found in the otolith that's likely drifted away from a little far away. However, fossil otoliths do come with their own challenges. One is that many fish species migrate in response to temperature changes, making it difficult to pinpoint specific environmental conditions they were exposed to. So it kind of shows, like, if we're seeing a bunch of fossils in the record and we're like, oh, gasp, the ocean ran out of water, or sorry, the ocean ran out of oxygen, or something like that, it's more than likely the fish just moved to another place that was better suited for them, rather than them changing their entire chemical structure and biological processes, right? Additionally, fossil assemblages often accumulate over a long period, mixing signals from different time frames and potentially complicating climate reconstructions. So they are really useful, but they need to be taken with a grain of salt because otoliths can move around, right? They're really small. They're usually less than 1 mil, well, depending on what size fish we're looking at, could be like as small as 1 millimeter or even smaller, and then they could be as big as 4 to 5 centimeters. So just depends. And then lastly, I do want to discuss how cool otoliths, fossil otoliths, can be if we can find them. So this is a study done in the Arkadelphia Formation, and they found over 2,100 otoliths from 19 species. When they quantified this data, they found that most of these otoliths were from cod-like fish, and this shows an evidence of cool water influences in the ocean. So this study was, sorry, I should have mentioned, this study was done in Western Interior Seaway, but down in Kansas. So different, again, Western margin or more southern, right? And it showed that, yeah, cool water influences are being entered into the area. And then when they also did some isotopic work, they suggested a shallow marine environment of less than 20 meters of depth. When I was discussing the Western Interior Seaway yesterday, we were kind of guessing ours was 200-ish meters. So we're a lot deeper up here in Manitoba than they were down in Kansas. But these findings did allow the researchers to refine paleo shoreline reconstructions and link fish species to specific areas in past environments. So it does grow the assemblage of the fish that were found in specific areas. And it's a great example of how fossil otoliths, when preserved under the right conditions, can act as detailed paleoecological archives. Now, that was a little rambly, but hey, I love otoliths and I love fish, so I'm always happy to do it. To conclude what I discussed today, it's modern otolith studies are crucial in understanding the impacts of environmental pollution on biomineralization processes. That's what I'm doing. By analyzing fossil otoliths, we can also gain insights into the climate and environmental conditions of the past ecosystems. In my case, Western Interior Seaway. So in the next steps of my research, I'll be focusing on microfossils from the Favell Formation and continuing to look through them digging my little hands around and hopefully finding some otoliths, because forams exist, so maybe otoliths do too. Or maybe they're just really small. I don't know. And then yeah, I'm gonna follow up with Bruno and see how he's gonna test the diagenetic alteration, because that's more along the lines of his thesis, and we're gonna see how we can kind of maybe collaborate and figure out what's with the lack of otoliths in the Pierre sham. Yeah, thank you very much for coming. I do just want to give out a couple of shout outs. So thank you to my supervisors, Kirsten and Michael, for listening to my silly questions and honestly just being such great people to bounce ideas off of. And a very special thank you to my friend Melina. Thanks for possibly nominating me for this. And thank you for always being there whenever I need to go get a coffee or just like, again, bounce ideas off of, because I work in a paleo lab, but there isn't a bunch of fish people around. So I will love all the fish people I can get. Yeah. And thank you especially for PaleoBricks for hosting me today. It's been such a pleasure. I had so much fun putting this together. Here's a list of my references, and I'm happy to share these with whoever wants to discuss or, I don't know, look into otoliths more. Thank you very much for your attention, and I'm, yeah, happy to take questions now.
Great. Thank you so much, Mariam. It looks like we have a couple of questions coming in already, and if you are going to DM us a question, please be sure to specify if you'd like to be anonymous or not. Here, first we have a question from Jose. Jose is wondering how vital effects are considered when doing chemical or isotope analyses on otoliths, or is it assumed that the otoliths are formed in equilibrium?
So wonderful question. Sorry, vital effects? Was that the first part of that?
Yes, we're popping it in the chat now.
Okay. Yeah, no, I actually don't know much about that. I would assume that, well, otoliths are formed in equilibrium, but I'd love to chat about what vital effects means. Maybe I'm not much of a biologist as I thought. The physiology aspect of things they're hard, and we don't have a physiologist on hand. So it's really complicated how otoliths are formed, and we're trying to figure that out every day. Especially because there are some elements that will come in through different channels, and then some that kind of sneak in there. Like I recently learned that selenium can sneak in through the calcium channel. So I'm not exactly sure, but that's a wonderful question.
Great. We have another question coming from Tom, who is wondering whether diagenetically altered otoliths can be identified and separated in analysis.
Yes. Yeah, they absolutely can. We have one that is kind of, it was Aaron Kilmary's paper. They said it was an otolith because it looks kind of odd and it looks a little otolith shaped. When I looked at it again, I was like, I don't know if that's an otolith, but it's very, very possible that it's an otolith that went under some alteration, right? So if parts of the otolith kind of dissolved away, that weird thing is the only thing we're left with, and perhaps everything else was aragonite, and that got dissolved, and then we're only left with the calcite parts. So yeah, they will be separated. We're not going to include those, but I don't know how confident I am that we will find otoliths. I'm really hopeful, but I don't know. I don't know.
Of course, I mute myself. That's really interesting. I have some questions about that, those diagenetic alterations as well. We have a question from, I don't know if it's Giannis or Janice, but they're saying, interesting talk, thank you. I have a question to the existing otolith data, both for recent and paleo environments. Are there usually data sets where you have several otoliths for the same time period that show similar signals, like pollution spikes? Using this, are there efforts similar to like tree ring constructions or other limnology where many records are aligned to a longer time series?
Yeah, very, very, very similar to the tree ring reconstructions. So for my master's specifically, I think I started out with 100-ish fish. And from that, I categorized them in different species. And right now, I think I have 3 or 4 species that I'm looking at. I ran my laser lines, and what I'm in the process of is figuring out if I can find spikes of similar elements in the same year. So step one is to look at my lines, step two, but also step one is to age them. Um, and a little bit of a sneak peek, I am seeing like some common spikes. So they're not spikes I expected. Like, sometimes the zinc will spike and the selenium will drop, and then sometimes the selenium will rise and the zinc will drop. So what I'm in the process of doing is seeing if that's common through all of my samples that I've run, and seeing if that's a species specific thing or if that's just because all those fish were found in the same lake, and if everything is spiking, let's say in 2012, then maybe there was something going on in 2012 that directly led to that spike.
That's really interesting. We also have an anonymous question asking, you mentioned that the area that you're studying is a pretty remote area with a history of mining. And they're wondering if there's a local community that's affected by the contamination from the mine and if they have like a stake in your research.
Yeah, yeah, they do. So Schist Lake is the lake that I'm focusing on. It's located just outside of the town of Flin Flon, which is Manitoba and Canada in general. It's like only southern part of Manitoba is really, really populated, and then everything else is a lot less populated. So it is a town, not a city. But yeah, so we are going up there this summer to do some field work, and we're directly collaborating with the local people of the town so that we're able to do our science research but not affect anything. But everyone I've talked to so far has been super duper helpful to help, because this is usually pretty non-invasive. Like, we're not taking thousands of fish out, and when we do, we kind of just need the head. So the rest of the fish can be used for food, and we're happy to let anybody who wants it have it. Do they want to eat it because it's a little polluted? It's a little debatable, right? They do rely on Schist Lake for food. So a lot of people will eat it, but um and recently, this past summer, the lake actually went on a selenium advisory, so they found really high levels of selenium in the fish mussel. So I wasn't able to get up there the last summer, but since then it's kind of been under an advisory where you're not supposed to eat more than one fish a month from Schist Lake. Yeah, so the community is affected, sadly, and I really want to do all I can to help out the community, even if that's providing them like the solid concrete proof that the mine has directly polluted the lake and that's bad and we should fix it. Like, bad.
No, that's so powerful. I'm also really curious what how the mechanism of that, like how you take the head and give the rest to be eaten.
It's like, yeah, technically, like if I'm looking at my head like it's a fish head, right? Like my ears are up here. If you
Right.
cut on my neck, I don't need any of that. Like I don't care about the liver. I don't care about anything. So you eat the rest of the fish. I only need the head so I can take the ears out.
That's awesome.
Yeah. We have
Um,
freezers full of heads.
I had a question as well, if I may. I am very much not a geochemist, so forgive me if this is a rudimentary question, but I'm curious if if we have issues in diagenesis with other sorts of biomineralized structures from other taxa turning into vaterite, or is that just an otolith thing?
It's like, so I've done very, very basic research on this, right? I've done some studies because the vaterite question is so intriguing. Like, coming from a biologist perspective, anytime we see an otolith that looks a little translucent or crystalline, they say that's vateritic, that's crystalline, we can't use that to age. So they just kind of toss them away. Coming from a microchemist perspective, we look at those otoliths through the TEM or we do extra diffraction. Sometimes they're not vaterite. So it's like there is something else going on there that's making the otolith look weird, look crystalline, look translucent, but it's not a different polymorph of calcium carbonate. And from what I can tell, like I'm thinking of like shells and any kind of ocean dwelling or shell dwelling animals. A lot of them, most of them, 95% of them are aragonite, and I don't think vaterite is very common there as well. Calcite is for different structures, but not, yeah, not the vaterite. It's supposed to be really rare, but we're seeing a lot more of it nowadays. Don't know why.
That sounds like a fascinating path for, further research. We have another question on diagenesis. We have a common theme here. Can you talk more? So you talked, a bit about the kind of the deep time potential, but can you talk a little bit more about, fossil otoliths and the effects of diagenesis on it? Are there any other than the vaterite and calcite changes that you mentioned, or how applicable, or how easily can we parse apart changes from very old otoliths?
So from what I can tell, there is like if we're saying there's 100 papers on otoliths, there's 2 on fossilized otoliths. So there isn't a lot. And I think when people find fossilized otoliths, they're not really doing research on the bad ones because there's thousands of good ones to be found. Um, like most people have no struggle finding otoliths. And I know of so many places where you can like just kind of you go on a field site and you just look around and you're like, oh, otolith, otolith, otolith. And here I am like begging for one. So I don't think there's been much done on the preservational potential of otoliths. Like they're just it's kind of a lacking area. Um, and then if we kind of so that's the paleo aspect of it. There's also an archaeological aspect because when early humans ate fish, same thing, like the body was disposed of. Otoliths stayed behind. They're not bones, so they stayed behind. We have a lot of these otoliths that we found from specific tribes that are like really old, and they're trying to find out if we can do stable isotopes on them, but because they were cooked, they don't know if that affects the otolith composition. So it just seems like there's a million questions out there and people kind of focus on 1 or 2 of them, and then the rest is a gray area. So if anybody likes fish, there's a huge potential there.
I love all the nuggets of potential career opportunities you're dropping here. This is amazing. You keep asking questions, or you keep answering questions that lead to the next question. So thank you. That's very convenient. So we have another question asking, you have this like really incredible specialized research happening already, but the person is asking, what lateral fields you could apply these methods to from otolith research. So maybe that is like archaeological stuff, or maybe it's other paleo things.
Yeah. So okay, so let's think. It's like 90% of the research on otoliths is done on aging and how we can better improve aging and how sometimes it's not even research, it's just word of mouth, or it's like we have aging labs out there to age fish and then look at population dynamics and look at migration patterns, and we look at find little areas where juveniles are being bred or breeding sites, right? All of that. And then there is, and I'm speaking from experience, the otolith symposium that I went to, it happens every 4 years. So from what I could see, most people were agers. 10% of the people were microchemists. And they're all coming at it from a biochemist point of view. So our project is coming at it from a geochemist point of view, and we're treating that otolith not like it's a bone, and we're just treating it like it's a rock that can tell us cool things about the biology. So a lot of the times, like I like to connect it to the fish, I like to connect it to the water, but that's because I like water. I like limnology, right? My supervisor, he doesn't like to think of it like a fish at all. He just treats it like a rock, or he treats it like a piece of aragonite that he's like, let's look at the chemical composition and let's look at the mineralogy in this. So that's already like the field I'm in is already a different than most of the other people that are doing this. Like I think yeah, we're one of the first people to do some of these methods on otoliths because oftentimes they're overlooked. And then yeah, archaeology has a huge part of that as well. And I know we have some archaeologists that we collaborate with constantly, and we just discuss, and we're always like, it's kind of like it's known. It's like if you find an otolith, we don't care what field you're in, let us know. We'll do something. Like, uh, yeah, I'm willing to collaborate with anyone. Like, I'm currently working on an instrument that was made for the semiconductor industry. And I was like, what's a semiconductor? Like engineering. I was like, what am I doing with my ear bone in an engineering instrument? It's so cool.
That's fantastic. Thank you for sharing. That's really interesting. I think we're going to wrap up questions there. Give you a break from all of the many fantastic questions. And so with that, for real this time, we want to say thank you for joining us. And please fill out the weekly feedback form. Join us again next week and stick around for tea time, where we will have this informal chat in a relaxed setting. But again, feel free to take a couple minute break and we'll see you in about 2 minutes.